Good afternoon, everyone, and welcome. I'm delighted to have you join us for our third presentation in the Osher Lifelong Learning Institute's 2026 At the U lecture series. As you have likely noticed, I am not Laura Peterson. I am Brianna Smith, the OLLI program manager, sitting in for the fabulous Laura Peterson as your facilitator. The Osher Lifelong Learning Institute at the University of Minnesota is a vibrant learning community geared toward learners aged 50 plus. Part of the College of Continuing and Professional Studies, OLLI belongs to a national network of Osher Lifelong Learning Institutes supported by the Bernard Osher Foundation. I'd also like to thank our sponsor for this series, the University of Minnesota Alumni Association. We acknowledge that the University of Minnesota Twin Cities is built within the traditional homelands of the Dakota people. It is important to acknowledge the peoples on whose land we live, learn, and work as we seek to improve and strengthen our relations with our tribal nations. Let me introduce today's presenter. Today's lecture is titled Immunotherapy, a Game Changer for Cancer Patients, and we are joined by Dr. Chris Pennell. Dr. Chris Pennell is a professor in the Department of Laboratory Medicine and Pathology and a researcher in the Masonic Cancer Center at the University of Minnesota. Dr. Pennell also co-directs the Institute for Engineering and Medicine's INSPIRE program and the Cancer Center's M-Ascend program. Dr. Pennell, welcome back to OLLI at the U, and the floor is yours.
Thank you very much. Thank you very much, Bree. Hopefully this works. Is that visible, Bree?
It is. Looks good.
Okay, so I'm good to go. All right. So thanks for the introduction. And again, I appreciate speaking with all of you. So I'm going to start with a story. This is a picture of Emily Whitehead, a girl from Hershey, Pennsylvania. This picture was taken on her 5th birthday back in May of 2010. 2 weeks after this picture was taken, Emily was diagnosed with a type of blood cancer called pre-B-cell acute lymphoblastic leukemia. The reason I'm showing you her picture is that Emily was the first pediatric patient treated with a novel type of immunotherapy called CAR T-cell therapy. And I've got a short video, maybe 2 or 3 minutes long, in which her parents explain her journey, and I'll play that now.
It was amazing. When she was born on May 2nd of 2005, she weighed 10 pounds, had a full head of hair.
When she came into our lives, she completely turned it upside down, but in a good way. She just has a ton of energy and likes to get up early and stay up late, and she's just been a blessing to us.
My name is Emily. My favorite color is purple, and pink and bluish greenish.
She's just a really creative little girl. She loves to draw and paint and do crafts. We think that she's just really strong and really brave because of what she went through. She went through 16 months of chemotherapy. She was supposed to go through a total of 26 months, and we got to 16 months of treatment when we found out that she had relapsed. The chemotherapy didn't work, and our hospital there told us that there wasn't anything else that they could do. And so they said that we could take her home and we would have a few weeks with her, or possibly a few months.
It even got to a point where Carrie had to stop doing research because the research showed that she wasn't going to survive. So I would tell her, no one has done a study on Emily yet. It's not an option not to have a positive outcome here, and it's going to happen.
Emily came to us in April of 2012. She had been in the hospital for nearly a year and was unable to get into remission. She was the first child enrolled on our protocol, and so we had a lot of doubts as whether the infused killer T cells that we made would be able to keep up with the rapidly progressing leukemia cells.
Some parents naturally would think of, I'm not sure if I just want my child to be a scientific experiment, but trying these experimental studies is what leads to breakthroughs.
I had read that immunotherapy was what doctors and researchers were kind of going towards, to use your immune system to fight cancer. The way that they explained it to us in kind of simple terms was that they would take her T cells out of her blood and that they would train them in a lab, or that they would send them to boot camp. then we're going to put them back into your blood, and those cells are going to find cancer cells in your body, and they're going to get rid of them. They had expected her to get a fever about 7 to 10 days after the T cells. She was on a ventilator for 2 weeks. But once she got on the ventilator, I think the doctors and researchers realized that it was because the T cells were actually working.
And my cell phone rang.
He looked like he was in a daze, and he was just staring at his phone. And I said, "What's wrong? There must be something wrong." And he said, "No." He said, "Dr. Grupp just called me, and he said the T cells worked." He said she's in remission.
It's been an amazing journey for me. It's been over 20 years that I've been working on this, so that's obviously very rewarding. There's still a lot to do.
So that's Emily's story. She's currently a junior at the University of Pennsylvania. That's her picture on the far right. She's been in remission for over 14 years. She would have died otherwise. And Carl June is a colleague of my colleague, and so I've gotten to know him. And he said that if Emily had died, the field would have ground to a halt, because for pediatric cancer trials, you don't want your child to be the second enrolled in the trial if the first child died. So it was fortunate for both Emily and the field that she survived. So I just thought I'd start with that story to show you the power of immunotherapy, because again, Emily was only given weeks to live after she went back into remission. So, I mean, when it recurred. So what I'd like to do is to just give you a quick outline as to what we're going to talk about. I have a couple of, I'm going to spend probably half the time on background just to put what I'm going to say about immunotherapy in the appropriate context. So we're going to start off with primer on cancer 101. What is it? How does it occur? How does it adversely affect us? Spend a little more time on immunology, just so you have an understanding of how we can exploit the immune system's natural functions to treat cancer. And then I'll go into specific types of cancer immunotherapy that are in the clinic as well as in the lab right now. So we're going to start off with a cell. We all start off with, whoops, sorry about that. We all start off as a single cell. Half of the information in that cell came from dad and the other half came from mom. So we start as one cell. We wind up at about 10 or 40 trillion cells in the average adult. And there are over 200 different types of cells, all of which originated from this original one. That's a lot of cells and a lot of growth that's occurred. Cancer happens when one of those 10 trillion cells goes rogue and doesn't obey the rules. And so what are the rules? The rules are to grow or divide, replicate, I'll use those words interchangeably, when you're supposed to. So one cell becomes two, two becomes four, and so on. And that's how that single cell ultimately yielded 10 trillion cells. But you're only supposed to do this when you're told to. Be where you're supposed to be. If you're a muscle cell, don't find yourself in the bloodstream. Do your job. Make the molecules that you're supposed to make, do what you're supposed to do. And then when it's time for you to say farewell to the body, die. So die when it's time to die. Cancers arise from rogue cells that don't obey those rules. But cancer is not a term used to describe a single disease. It's an umbrella term that describes hundreds of distinct but related diseases. And they're distinct because they arise in different tissues. I told you that there are over 200 different types of cells, 200 different types of tissues. They can arise from, cancer can arise in those different sites via a variety of different causes, virally induced mutated genes, which we'll talk about a little bit more later. And there are different prognoses. So when Emily was diagnosed with pre-B-cell acute lymphocytic leukemia back in 2010, her parents were devastated by the diagnosis, but they were heartened by the prognosis, because children with that blood cancer typically had about an 85%, 90% chance of surviving until adulthood. So even though it was a horrible diagnosis, they thought that Emily would be cured of the disease. As you saw, she was not, unfortunately. She fell into that 10 or 15% that were not susceptible to the therapy. But there are different prognoses for different outcomes. So that's how cancers are distinct from one another. But they're related by two key characteristics. One is this uncontrolled division or growth or proliferation. So they don't stop dividing when they're supposed to. That is the definition of a tumor. A tumor essentially means, it's Greek for swelling, and it means uncontrolled growth. So a wart is a tumor because the cells are growing uncontrollably. That's what causes the bump. But what distinguishes a benign tumor, like a wart, from a malignant tumor, like leukemia, is invasiveness, where cells spread locally or throughout the entire body. And this process is called metastasis. Meta means beyond, stasis is stillness. So this is a double negative, beyond stillness, meaning it moves. So a tumor can be benign or malignant. Tumor just means uncontrolled cell growth, but a malignant tumor is synonymous with cancer, and that means an uncontrolled growth that is moving either locally through the tissues in your skin or systemically throughout your body. So how does cancer adversely affect us? This is a picture of an individual with melanoma. The suffix OMA, oma, means a tumor. Melanocyte is a freckle, a pigment-forming cell. The pigment is melanin. So a melanoma is a malignant freckle or a malignant pigment-forming cell. And you can see that it's spread throughout this person's leg. Unfortunately, melanoma also metastasizes to the brain. And so these arrows point to these white spots, which represent metastatic melanoma growth. And so you can imagine as this grows larger and larger, it's compressing the healthy brain cells around it. And so it's compromising the normal function of this organ. And that's what can lead to death. So that's how cancer adversely affects us. And the way that I think about this is the 4 S's. So cancer cells are like weeds in your garden. They can grow more rapidly than healthy cells. So they can starve healthy cells for nutrients. They can compete those cells for oxygen. They squeeze them, compromising their function. And then also these cells, some tumors secrete molecules that are needed by the body in small amounts. But if you've got 10 billion more cells than you need, that excess protein can be problematic. So these are the 4 ways in which cancer adversely affects us. The next question is, well, how do these rogue cells go rogue? And the answer is, it's from the corruption or misuse of information, genetic information, or DNA. So DNA is an acronym for deoxyribonucleic acid. That's a mouthful. I'll just say DNA from now on. But you can think of it as a biochemical information storage and retrieval system. And the entire collection of a cell's DNA is called its genome. OME, this suffix represents the entirety of something. And a gene is a unit of information, a genetic unit of information. When I think about these processes, I like to use analogies to make myself understand them better. So some of the analogies for a genome, the entirety of the genetic information a cell has, include a computer's hard drive or an owner's manual or a cookbook. So if we use these analogies to describe a gene, one unit of information, one file on your computer or one recipe in the cookbook is equivalent to one gene. So this is information. And the product of this information is a protein, as opposed to a cookie or piece of paper that you print out from your Word document. So a gene encodes information that makes a protein, and this is the molecule that actually does the work. So again, taking that analogy, these are 3 separate cells from an individual. Each of these cells has the identical genetic information or the identical cookbook. We have 23 pairs of chromosomes. 23 came from mom, 23 came from dad. So you can think of these as chapters like 23A and 23B, for example. A gene is a unit of heredity, a particular recipe. All cells share common functions like making energy, making molecules that they need, and we call those housekeeping genes. So these are bits of information, recipes that all cells use. But the reason why a heart cell is different from a white blood cell is because those cells use different recipes in the book, and we call these tissue-specific genes, and they define what a cell will do. The entire book contains almost 13 billion letters, and the letters of the genetic alphabet are just 4 of them, A, C, G, and T. And every letter in this book has to be copied when one cell becomes 2. And what I'm painting is a picture where the odds are not in our favor. So on average, the average adult human makes about 300 billion new cells every day. New white blood cells, new skin cells, new cells in our digestive system. So if we make 300 billion cells a day, and each cell has got about almost 13 billion letters, that's 3.87 times 10 to the 21st, or roughly 1 trillion billion letters are copied every day in the body. If we put that into length, that works out to about over 400 miles of DNA is copied every day in the body, which is about 4.5 times the distance from the Earth to the sun. This is every day in our body. This is copying that's going on every day in our body. So with numbers that astronomically large, mistakes are going to happen. And this is how cancer arises. So I mentioned that cancers have 2 commonalities, undivided growth and this invasion or spread. They also have other what are called hallmarks of cancer. So, for example, here, number 6 on the right, they make their own blood supply because as they grow larger and larger, they need their own supply lines. So they pave new roads, if you will, that allow blood vessels to get in and feed them oxygen and nutrients. They use different sources of sugars and proteins for their growth. They don't die when they're supposed to die. And the one that we're going to focus on is number 6 down here. I don't know why that's 6. That shouldn't be 6. That should be 4. Anyway, it's evading immune destruction. So most cancers share these things. And again, to think of it in pictures, I think of these hallmarks as barriers that we have evolved to prevent cancer from happening. And so in this diagram, there are healthy cells here that are in green, but I've highlighted 2 of them that are in blue. And so let's say these cells need to divide for whatever reason. So that one cell becomes 2 healthy cells. But this one, when it divides, one of the progeny has a mutation in it. And that mutation allows it to go over the first wall. And then when it divides, another mutation occurs that allows it to acquire a second hallmark of cancer, and so on and so on and so on down the list. And so this process typically takes years to occur. But it's these mutations or changes in information that allow cells to overcome the barriers that we have evolved to prevent a tumor from happening. And I like to think of this last wall as the immune system. And so with this background about cancer, we're going to get into a little bit about how the immune system functions before I just talk about immunotherapy. I'm jumping ahead of myself here. A little bit more about the biochemistry of cancer. So how can a mutation in a recipe change to lead to a change in a cell's behavior? Well, as I mentioned before, genes just have information. So altered gene products lead to altered proteins. These are the molecules that do the work. And so, for example, here, if we look up here, these triplets, AAA, GGG, ACT, these are letters in the DNA alphabet. 3 A's in a row are translated from the language of nucleic acids to the language of amino acids in a protein. So 3 A's equals a lysine, which is a particular type of amino acid that is one of the 20 or so building blocks of proteins. GGG encodes glycine, ACT encodes threonine, and so on. Lysine is a positively charged amino acid residue. Aspartic acid is a negatively charged amino acid residue. So if these were part of a protein, you could see that the positively charged residue would attract the negatively charged one, and these intervening Lego bits, if you will, pieces of Legos or amino acids, they would loop out and form a loop. And this loop could be functionally important. It may serve as a docking site for another protein to bind. So what happens if a mutation occurs? Well, if that first A in the AAA triplet is mutated to a G, it changes from a lysine to another aspartic acid. This was a mistake. Sorry, there should be ASP here. So now you've got two negatively charged molecules that repel one another, and you've destroyed that loop and that function. So that's how a single mutation can disrupt a protein's function. Okay. So how does all this happen? So how do we get cancer? It boils down to three different ways. Age, the accumulation of mutations over time. I was born in New York City, was raised on Long Island. We went to the beach quite a bit because my dad loved to swim in the ocean. I'm fair-skinned. When I was a child, parents weren't slapping on sunscreen. And so I got sunburned quite a bit. And as a result, I've had a couple of skin cancers that have been luckily caught in time. But probably the first mutation that led down that pathway occurred when I was 4, 5, 6, 7 years old. But it took another 30, 40 years for the additional mutations to occur to make that cell become malignant. And so with that, it's logical that cancers primarily strike older adults because we've been around longer than a young child, and our cells have had more time to accumulate these changes in DNA. So this graph on the x-axis shows age in 5-year increments. And on the y-axis is the daily adjusted cancer rate per 100,000 people. So you can see it's pretty flat until you hit your early 30s or so, and then it increases pretty rapidly as we get older, just because cancer typically results from the accumulation of mistakes over time. The second way is just bad luck. You can inherit a mutated gene from one of your parents. When your cells are copying the DNA, a mistake may not be caught and corrected. You can also be unlucky if you're accidentally exposed to things in the environment that can cause DNA damage. So a classic example of this was the Chernobyl nuclear reactor meltdown in what is now Ukraine back in the 80s. That meltdown released a plume of radioactive gas that floated over Europe in a southwesterly direction. So children that were outside playing inhaled these radioactive isotopes, and they have a higher risk of cancer. They weren't doing anything except playing, right? Certain viral infections. And then the last is your lifestyle. So if you intentionally expose yourself to something we know can increase your risk for cancer, you may get cancer more than somebody else. And the classic example of this is cigarette smoking, right? So of these three, the only one that we can control is our lifestyle. When I give talks like this to students, I don't get up on a soapbox and say, don't smoke, don't do this, don't do that. I just say, here are the data. Hopefully everybody will live to be a ripe old age. Not much you can do about bad luck. The only thing you can control is your lifestyle. So you make those decisions. Okay. We're going to switch now a little bit and talk about how you deal with cancer if it becomes clinically apparent. And cancer therapy has evolved over the years. So before the 1800s, surgery was the only option. At the beginning of the last century, radiation therapy and traditional chemotherapy came into play. And at the beginning of this past century, we're getting more and more precise with our therapies. So there are pharmacogenomic approaches called precision therapy, and then there's immunotherapy, which is obviously what we're going to talk about. And what's driven this evolution is specificity. So if you can develop a drug or a treatment that targets only the malignant cells and not any healthy cells, you have a much better chance of eradicating the disease than you would otherwise. Because what happens with almost every drug is that you reach what's called the maximally tolerated dose, meaning you can't give any more of that drug to a person without killing them from the drug rather than the disease. And so it's this specificity, which is the holy grail of cancer therapy, that's driven the evolution of this. So what is immunotherapy and why is it useful? Well, as the name implies, it uses the immune system to treat a disease such as cancer. And it's been a game changer for cancer because it has these 4 characteristics. It's exquisitely specific. So the immune system can distinguish between 2 molecules, 2 proteins, for example, that are identical except for one single amino acid residue. So you can have 2 proteins that are 1,000 amino acids long. If they differ by just one due to a mutation, the immune system can recognize that. The immune system is incredibly potent, as you'll see in a few minutes. It also has a memory. So if the disease goes into remission, but then it recurs, the immune system has the ability to eliminate it once again. And then the last thing is adaptable. I didn't show you this in the cancer hallmarks, but one of those hallmarks is genetic instability, meaning that cancers acquire mutations at a higher rate than their healthy cellular counterparts. And the immune system can basically hit a moving target like that. Okay, so here's Immunology 101. The immune system functions to maintain balance or equilibrium or homeostasis in the body. And when that homeostasis is perturbed by injury or infection or disease, the immune system becomes activated. And in a healthy adult, under normal physiological conditions, the end result of immune activation and then subsequently turning off the immune response is a return to this balance or this homeostatic healthy state. So the wound is healed, the infection is resolved, the disease is eradicated. One of these homeostatic functions of the immune system is called immune surveillance. And this means that the immune system is constantly eliminating nascent tumor cells before we even know we have them. Okay, so this is just a little diagram. This didn't show up very well, but those immune cells should have moved across the screen to eliminate the tumor cells. This is happening probably daily in our bodies because if we're making 300 billion new cells, it's likely some of those are going to have mutations that can be recognized by the immune system and eliminated before the cell can be problematic. With that definition, then, clinically apparent tumors have somehow escaped immune recognition. And I think of this as a wall that the tumor cells have erected between themselves and the immune system. And so even though immunologists have long recognized the specificity of the immune system would be ideal to treat cancer, we couldn't exploit the immune system to treat cancer because we didn't understand how that wall was built. We couldn't overcome it. But it's been due to research over the last 20 years or so that we've gotten a much better understanding of the bricks and the mortar that are used to create that wall. And with that understanding, we can either reduce the height of the wall, destroy the integrity of the wall, or basically just elicit such a strong response that it crashes over that wall and it can kill the tumor cells. But again, it's been this wall that tumor cells have used to evade immune recognition that stymied the use of immunotherapy for over a century. So as I mentioned, the immune system maintains homeostasis. One of its functions is to protect us against the sea of pathogens, right? And so as they enter our body, the immune system can recognize them as being foreign and eliminate them. But how does this work? How does the immune system do this? And it does it through the actions of white blood cells. And there are many different types of white blood cells, but we're going to focus on 2 types called B and T cells, because these are the ones that have that exquisite specificity that I've alluded to. So this is just to show you that there really are white blood cells, and they are white. So this vial on the left underneath the word before represents blood that's just taken from an individual. This is after it's been centrifuged. So the components are separated based on density. And at the interface between the liquid phase of the blood or plasma and the red blood cells are the white blood cells, and they comprise about 1% of the volume of the blood. And it's these cells that keep us healthy. As I mentioned, there are 2 types of cells, immune cells, white blood cells, that have this exquisite specificity. One type are called B cells, and these develop in the bone marrow. And the way that they function is they secrete molecules that are called antibodies. So these molecules are soluble, they're floating around in our blood and in our lymphatic tissues. And they protect us from extracellular attacks. So things that are outside of those 210 different cell types that I mentioned. These would be bacteria or viruses that are floating around our body. Anything that's outside of the cell. Okay. And this is just a short video showing how they work.
Block, flag, deliver. How monoclonal antibodies treat cancer. Antibodies are molecules that our bodies make to help fight germs. Monoclonal antibodies are similar molecules that are made in laboratories and are used by doctors to find or treat cancer and other diseases. This video explains what monoclonal antibodies are and a few ways they're used to treat cancer. Antibodies are Y-shaped molecules that attach tightly to a target. They're very specific, meaning that each antibody attaches to only one target. An antibody and its target fit together like pieces of a puzzle. In the laboratory, scientists can make many identical copies of a monoclonal antibody that can attach to a specific target, such as a molecule on the surface of cancer cells. These monoclonal antibodies can block molecules cancer cells need to grow, flag cancer cells for destruction by the body's immune system, or deliver harmful substances to cancer cells. This makes them a valuable type of targeted therapy for treating cancer. For example, a monoclonal antibody called trastuzumab attaches to a molecule called HER2 on the surface of some cancer cells. Blocking HER2 keeps it from sending signals the cancer cells need to grow. Another example involves VEGF, which is a molecule that makes blood vessels grow. A monoclonal antibody called bevacizumab blocks VEGF. Blocking VEGF stops the growth of new blood vessels that the tumor needs to survive. A third example is the monoclonal antibody pembrolizumab. Pembrolizumab attaches to molecules called immune checkpoints on immune cells. Blocking immune checkpoints helps the immune cells kill cancer cells. Other monoclonal antibodies treat cancer by flagging cancer cells for destruction. For example, when the monoclonal antibody rituximab attaches to a molecule called CD20 on cancer cells, it acts like a flag for immune cells. The immune system sees this flag and destroys the cancer cells. Some monoclonal antibodies fight cancer by delivering drugs, toxins, or radioactive particles to cancer cells. For example, brentuximab vedotin is a monoclonal antibody that is linked to a chemotherapy drug. When the antibody attaches to its target on cancer cells, it delivers the chemotherapy drug, which kills them. Cancer researchers are continuing to investigate new ways to use the precision of monoclonal antibodies to treat cancer patients.
One of the reasons I showed you that video is that pronouncing those names always trips me up. So I wanted to save myself the embarrassment of trying to pronounce some of those names. But if you noticed, almost all of them ended with MAB. That stands for M is monoclonal, AB is antibody. So if you're watching television and you see something with an MAB at the end, you know it's an antibody. That's derived from an antibody-producing cell called a B cell. Another type of immunotherapy involves vaccines. And the way that vaccines work are to increase the frequency of a cell type that can make a monoclonal antibody, for example, specific for the virus or a tumor cell in this case. And so all of these different colored balls on the left of the wall represent T and B cells with different specificities. They recognize different molecules. So if you come in and you inject a vaccine, you are expanding those cells specific for the molecule that's on the surface of the tumor cell or the virus and can eliminate it. And there are several different types of cancer vaccines that are available right now. They come in two flavors, if you will: prophylactic, which means preventative, and therapeutic means to treat a cancer. So prophylactic cancer vaccines are mainly ones that are specific for viruses that we know can cause certain malignancies. So subtypes of the human papillomavirus cause almost all cervical cancers. So there are vaccines that can be given to children, prepubescent children, that will guard against this infection. So if you eliminate the HPV when you get infected, it doesn't have 10 or 20 or 30 years to accumulate mutations in an infected cell that could lead to cervical cancer. There are other types of viruses like hepatitis B that are associated with liver cancers. And again, if you can prevent the infection of the virus, you can minimize the risk of getting liver cancer that's caused by that. There are also some therapeutic cancer vaccines that are given to individuals with a cancer, but they are more difficult to elicit a good response because you're trying to elicit an immune response in an immunosuppressed person. And so these prophylactic cancer vaccines are much more effective because the individual is healthy and is not compromised by either the treatment of their disease or the disease itself. One of the antibodies that was mentioned, nivolumab, is what's called an immune checkpoint blockade. One of the ways in which this wall is erected is that tumor cells have evolved ways they've figured out, if I can be anthropomorphic, how to turn off an immune response. And so they put on their surface a protein that engages essentially a molecular brake on a T cell or a B cell, and it shuts that cell down. The Nobel Prize was given in 2018 to Jim Allison because he identified this molecule, and the leap of logic that led to his Nobel Prize was that he identified this molecule in a viral setting. He then thought, I wonder if tumors have figured this out. So he made an antibody that blocked the brake and gave it to mice who had a tumor that was not recognized by the immune system. And once he put in that blockade antibody, the immune system was able to eradicate the disease. Jimmy Carter was treated with this back when he was 85, and it allowed him to live for another 15 years. So this basically allows the immune system to, it allows us to reduce the height of the wall or the integrity of the wall. And these are some of the antibodies that are used for this checkpoint blockade. And this is hugely successful. The other type of white blood cell that has this specificity are called T cells. And they're called T cells because they develop in an organ called the thymus. And in contrast to B cells, T cells recognize what's going on inside the cell. So if a cell is infected with a virus, that cell basically turns into a viral producing factory. If you can kill the factory before it releases all of the viral particles, you're going to be better off than if the cell had ruptured and released thousands of viruses after being infected. So what the T cell does is it's constantly scanning the surface of a cell for bits of protein. And it's learned in the thymus to ignore our own protein self. So it doesn't react to anything that we make typically, but it does attack cells that make something non-self or foreign. And this can be a viral protein, or it could be a cancer mutated protein. And so, sorry about that. This is how they recognize a virus. So if a virus infects a cell, the viral proteins are released. Then they're shuttled to the cell surface on a platform. And now this viral fragment is presented to the T cell. And if the T cell has a receptor that can recognize that, it can become activated and then kill the virally infected cell. But different T cells have different receptors, which is what allows us to respond to almost anything the universe can throw at us. So the frequency of any one T cell specific for any one viral protein is really low, which is why it takes a few days for the immune response to clear an infection, because it takes a while for that one T cell to find the virally infected cells and then become activated. This is a movie about how these T cells work.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explore the surface of the target.
So now the T cell is looking for something foreign, a mutated
Cytotoxic T
protein.
cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell. These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate.
Those are the types of cells that were given to Emily. So what happened in Emily's case is that they removed her white blood cells, not all of them, obviously, but some. And then Carl June and his colleagues genetically engineered those T cells to recognize something on the surface of the tumor. So even though each T cell by itself was born to recognize something different, through genetic engineering, that innate specificity was overridden, and now they were converted into cells that all recognized the tumor. And so you can think of this as like a tsunami breaking over a seawall. And with that many, and those cells were grown up to billions in numbers and then put back into Emily. And she would have died otherwise without this. So we can modify cells in the lab. The other thing that we can do is we can use what are called immune cell engagers. And you can think of these as molecular bridges. So as I've mentioned a couple of times, each T cell has a receptor, let's say CD3 here, that can recognize something on a tumor. But what we can do is we can add what's called, this is an antibody with two different arms. So one arm of the antibody binds the receptor on the T cell regardless of what this receptor was born to see. And the other arm binds something on a tumor. So it bridges the T cell and the tumor cell, and it's basically telling the T cell to kill this, even though the T cell would not recognize it without this molecular bridge. So these are the take-home messages that I wanted you to take home. Cancer arises from the corruption and misuse of genetic information, DNA. It's defined by uncontrolled cell growth and spread. Cancers occur from old age, bad luck, and your lifestyle, or a combination of those. Cancers are adversely affect us because they can outcompete healthy cells for food and oxygen. They can compromise the function of healthy cells, and they can secrete molecules that just perturb normal functioning. Emerging therapies are driven by specificity, and immunotherapy is a game changer because the immune system has evolved over millennia to be specific. This is just an idea to give you an idea as to how big business this is. So we're down here at 2025, 2026. So it's over a $120 billion market for the US, and it's anticipated to double or triple in the next 10 years, just because of the potential of this. And what I'm going to do is I'm going to end with a story of Emma Dimmeray. Emma is a University of Minnesota grad from the College of Art and Design. And when she was 23 or 24, she was diagnosed with, stage 4 colon cancer, which was devastating because she was such a young person. She went through 10 years of treatments, multiple treatments, and they all had failed. And so 2-3 years ago, investigators here at the university took a biopsy of her tumor, teased out the T cells that were in her tumor, and they were being shut off by this immune checkpoint. But those antibodies that I mentioned before didn't seem to work for whatever reason. So these investigators here at the U, Emil Liu, Bo Weber, and Brandon Moriarty, identified an intracellular, not on the cell surface, but something inside the cell that was serving as a brake. And through genetic engineering, they eliminated the gene that encoded that brake in Emma's T cells. Then they put those T cells back into her, and she's been in remission for 3 years now. So it's a brave new world. We're combining genetic engineering with immunology, protein biochemistry to try to treat malignancies that are resistant to standard chemotherapy and radiation therapy. Okay. And with that, I'm going to stop, and hopefully I can answer questions that you all have.
Thank you, Dr. Pennell. That was such a fascinating talk, and these stories that you provide of the successes of these therapies is really quite uplifting and exciting, I know, for me to hear, I'm sure for others. We have a number of questions in the chat that I will, or in the Q&A, excuse me, that I will get to. And for those in the audience, please send your questions to the Q&A, and we have lots of time for discussion, so we can take more. Our first question invites an international perspective and is wondering if immunotherapy treatments is also kind of the game changer in other countries, or if there are other treatments being taken on in other healthcare contexts, say in Europe.
Yeah, for sure. It's certainly not focused on the United States, or it's not exclusively used in the United States. So yeah, it's as you can imagine, countries that are more affluent have greater access to this. Many of these therapies are incredibly expensive. So the type of therapy that was used for Emily Whitehead, the little girl, cost anywhere between $300,000 and $500,000. So they're not cheap, but individuals were working on, and one of the reasons is that they are so specific for that person. So folks here at the U and other places are trying to work on off-the-shelf reagents, like that bridge that I mentioned. That bridge can be used in anybody as long as they have the target antigen on their tumor that one of the arms is specific for. So off-the-shelf reagents are trying to be developed, but it's a long process.
Of course. And just a reminder to our audience to direct questions to the Q&A. Since this is a webinar, we can't take your raised hand questions, but through the Q&A, I will do my best to get to all your questions. A question about supplements and thinking about what we can do for our immune systems. Is there definitive data supporting use of supplements like lysine to support the immune system?
Yeah, there are. In fact, that's funny you should mention that. I just was reading a paper yesterday about that. And the way that it works is that so for old folks like me, my T cells don't work as well as my grandsons, for example, just because the term is exhausted. And there's a molecular definition of that. But supplements like leucine and lysine can partially reverse that exhaustive state to make the cells more reactive, if you will. And so, yeah, there is solid evidence for some. I don't know them all other than this one because I happened to read that paper yesterday. But yeah. And the key thing, like anything else, is, if you do things in moderation, if you exercise moderately, those things will help. Obesity is one of the causes of cancer, and one of the reasons is that there's a constant adipose tissue causes low level of inflammation. And inflammation is one of the ways that the immune system uses to kill pathogens. And it does that by eliciting superoxide radicals that damage DNA. So if you have low levels of constant inflammation, you're exposing your DNA to constant, sources of damage. So those are, yeah. So that's another thing, yeah.
Someone is wondering about the difference between a TKI using targeted therapy and immunotherapy.
Yeah, so, and I am a mouse doctor, so I'm if people ask me about different drugs and doses and stuff, I'm useless, okay?
Yep.
But theoretically and conceptually, I can probably help. So when a T cell recognizes something on a tumor, that recognition event starts a cascade of signals that goes to the recipe book and opens up different recipes to turn that cell into a killer. part of that cascade is a molecule called tyrosine kinase. TKI is a tyrosine kinase inhibitor. And so you can use that inhibitor to stop that signal cascade. But those cascades are also used for a cell to grow uncontrollably. So depending upon the tyrosine kinase, there are different inhibitors because they all have slightly different shapes, and you need different drugs to plug up those sites. So it's basically the gas pedal for a cell to grow, and the inhibitor takes the foot off the gas. And so you can combine these TKIs with immune therapy as well. So there's a lot of combination therapies that are ongoing. And combination therapy is probably the way that the field will ultimately mature, because if you're targeting one particular molecule and the tumor turns off that target, now you're out of luck. But if you come in with 2 or 3 or 4 different therapies that target different molecules, it's very unlikely the tumor cell will turn off all of them at the same time.
And is that work already underway? I was
Oh,
kind of
yeah.
curious to ask that question, like immunotherapy game changer, there's got to be a what's next to kind of building on it. Is that?
Yeah.
Yeah.
The issue is with, so what's available in the clinic is about 10 years behind what we can do in the lab. And the reason for that 10-year gap are clinical trials. So if you come up with a new clinical approach, you have to first show it's safe, then you have to figure out how much to give, and then you have to compare it to the current gold standard of treatment in the field. And all of that takes a lot of time and a lot of money. And if you're combining 2 or 3, that makes it even more difficult because you have to look at them individually and then in the different combinations. So yeah, what we can do with mice is a lot better than what we can do for people.
Of course. Well, there are a number of folks curious about the applications of immunotherapy beyond cancers, whether this is going to being broadened out to address other diseases such as autoimmune disease. I know this isn't your specialty, but any awareness for this kind of broader application?
Yeah. So in fact, the type of therapy that was used to treat Emily kills antibody-producing cells. So it targets healthy B cells, which make antibodies, as well as malignant B cells like B cell leukemia, which is what Emily had. So even though she's a junior at Penn now, she doesn't have any B cells in her body, so she's immunocompromised. She has to get doses of antibodies pulled from thousands of people a few times a year. There are autoimmune diseases that are caused by B cells, multiple sclerosis, and a few others. So again, I was just reading a paper a week or so ago about this patient who had 3, poor woman, had 3 different types of antibody-associated autoimmune problems. She's been treated with this same type of immunotherapy, and now she's disease-free because the source of those autoantibodies, the antibodies that react itself, have been eliminated. So yeah, it is. And the immune, like every system in the body, it's a balancing act, right? So you activate the immune system, it gets rid of something, but if it stays active, it's going to cause problems. So then you have to shut it off. So it's trying to achieve that balance, which is really, really tricky.
Thank you. It seems like folks have been reading about other kind of approaches to cancer treatment that would love to kind of hear you weigh in on. Someone just reading about whole genome sequencing in ARP magazine. Someone wondering about DNA therapies. Are these different, or are these kind of in harmony with immunotherapy?
Yeah, they are in harmony.
Okay.
So for Emma Dimmeray, the woman that I showed at the very end, what the investigators here did was to extract DNA from healthy tissue of hers, extract DNA from the tumor tissues, and then compare the sequences. And by doing that, they were able to identify the mutations in the tumor. Several of those mutations could be targeted by the immune system. And so what they did was they translated that genetic information of a mutation into a mutated protein and used that to stimulate her immune system in the laboratory. And then when they stimulated those cells, then they put them back into her. So that was a combination of genome sequencing, molecular biology in the lab, immune cell activation, and then putting them back inside of her. So yeah, so That's one explanation. Other explanations are trying to identify the source of a disease. So sickle cell mutation, for example, is caused by a single amino acid residue change in hemoglobin. And that can be corrected through genetic engineering. And so some individuals now will have their bone marrow removed because the bone marrow is the source of all blood cells, white and red. That genetic mutation can be corrected, then they can kill all of the bone marrow cells and put the corrected ones back in, and now the person is free of disease. So yeah, it's phenomenal how much the field has evolved since I started 40 years ago or whatever it's been.
Right. And how much is this being adopted by, an oncologists, seeing patients, determining a course of action? Are they is there what is that line of communication from the research scientist to oncologist to patient like?
Yeah, that touches at what's called the town and gown
Okay. Yeah.
collaboration, right?
Yeah.
And many of those monoclonal antibodies that I was talking about before are available in the clinic. So an oncologist in White Bear Lake or wherever can certainly prescribe that and use that. That's okay. It's more of these the experimental ones that are not FDA approved is where it gets a little bit where the relationship has to be strong. So if somebody in northern Minnesota, an oncologist in northern Minnesota has a patient with a particular disease, they'll treat them with the standard of therapy for that disease first because that's the way to go. We have the most information about that. And if they are resistant to that therapy, then she may contact somebody here at the U and say, I've got this person, I don't know what's going on. And so then that collaboration exists. But ideally, what happens is that the person would come to a tertiary care center like the U, they're treated, but then they're put back into the hands of their oncologist because that person knows them the best, right? So yeah. So for the FDA approved ones, there's really no interaction between us and them, but it's for these newly emerging ones. And in fact, I think that was what Emma went through after failing, literally a half a dozen types of therapies. Her oncologist, contacted folks here at the U, and then it went from there.
Mhm. There's a lot of curiosity about what we know about the patients that have been most successfully responding to immunotherapy. Is there something about their blood type, some kind of something already existing in the immune system that gives them a leg up or makes them more successful in their
Yeah,
health?
so there was a fascinating study published a couple of years ago that looked at that exact question. So they looked at individuals that responded to this monoclonal antibody checkpoint therapy, and they looked at those people that did not respond. And the only commonality they found was what the bacteria were in their guts. So it was your microbiota, it's called, in your gut, which obviously is reflective of your diets and your eating habits and that sort of thing. And so at a molecular level, what they found was that in the people that responded well, there were bacteria that enhanced those immune responses and bacteria that made soluble molecules that removed the brakes that tumors could put on. And the converse occurred in people that were non-responders. They had a preponderance of bacteria that made things that enhanced the brake and removed the gas, if you will. So other than that example, I don't know of particular mutations in the DNA that will allow one person to respond better than another. But having said that, the second type of targeted therapy that's being used is called pharmacogenomics. And that, as that name implies, it's a combination of pharmacology and genomics. So in that case, what is done is that your healthy DNA and your tumor DNA are sequenced, and then the enzymology that you have is compared. And so sequencing your tumor DNA allows them to pick what drug is going to be best, what tyrosine kinase inhibitor, for example. And then by sequencing your healthy DNA, they can choose among 4 or 5 different TKIs which one they think you're going to respond to best based on your own personal biochemistry. So those are the sorts of things that are happening, and that's beyond my pay grade.
Incredibly specific, though, as you're talking this precision. That's
Yeah,
really
Yeah,
Really incredible.
yeah, it is. It's phenomenal. But that microbiota was just fascinating to me, you know.
Absolutely. So a question there, are the cancer researchers, are they digging into that aspect, the gut microbiome, food as medicine, or is that another camp of researchers, and are you talking to each other?
20 years ago, it would have been definitely different camps, and folks like me would have poo-pooed the other folks.
Uh-huh.
Now
Uh-huh. Yeah.
I've learned, the error of my ways, and everybody's talking. So, we have a colleague, Alex Koritz, who years ago was talking about what's called FMT, which is fecal material transfer. It's basically taking poop from one person and putting it into another. And that worked incredibly well for a type of infection called C. difficile, which a lot of patients undergoing surgery would get, and it caused diarrhea. It was unresponsive to, intensive antibiotics. But if you just gave somebody, you know, fecal matter from a healthy individual and gave it to the diseased individual, they were cured within like 4, 5, 6 days. It was unbelievable. And so he would give us these talks where he would show blenders of poop that were like, Alex, don't do that. But now they've developed a pill. So they've identified, the good bacteria and gotten rid of the bad bacteria. And so, yeah, but this has been a long process. To answer your question, yes, we are talking to people. Yes, there are dietitians in the cancer center. Yes, there are a lot of these things. In fact, there was just a clinical trial that was done by a colleague of mine looking at the effects of watercress on tobacco carcinogenesis. And it was shown that consuming watercress reduced the risk because it dampened down some of these reactive molecules that were being made. So yeah, it's fascinating. Yeah.
I got to figure out where to buy watercress these days.
Don't smoke is probably the better
Okay,
way to
right.
go.
That's the better route. You're right. You're right. Good point. Um, Question about side effects. We're all quite familiar with the side effects of chemotherapy. Do we see negative side effects coming with immunotherapy? What's
Yeah,
like?
yeah. It's difficult to make a blanket statement in biology is not a good thing. But having said that, pretty much every therapy has a good side and a downside, right? And so in the case of Emily, she had acute and chronic morbidity. So the acute problems were what's called cytokine release syndrome. And what that means is that when they put billions of these killer cells into her body and they found billions of tumor cells, it led to a systemic inflammation. And that could be problematic because you could destroy the lung tissue and other things like that. So we have a good idea as to what's driving that, and so we can dampen that down. The other type of acute toxicity is neurotoxicity in this type of therapy. And that was totally unanticipated. And it affects about half of the patients treated with this type of CAR T cell therapy. And the symptoms range from mild disorientation to fatal brain swelling. We don't know how it occurs. We don't know who's at risk. And so in my lab, that's what we're doing. We've developed a mouse model in which the mice get neurotoxicity. And so we're trying to understand how it happens with the long-term goal of preventing it. So those are the acute short-term problems. The long-term problem is that she doesn't make antibodies. And so she's going to have to be supplemented with antibodies for the rest of her life
Wow.
because there's no off switch for these cells. What is ongoing now in clinical trials are these genetically engineered cells are being engineered with an off switch or a suicide switch that can be activated once the disease is eradicated. So then you can get rid of the cells and then hopefully the antibody producing cells can come back.
Wow.
Yeah.
Incredible.
Yeah. But if you drink enough water, it will kill you. So it's a dosage effect, right?
Right.
I mean, literally, it could.
Yeah.
So,
Yeah.
there are always side effects. And what you try to do is minimize the side effects, keeping in mind that the person would have died otherwise had they not had this treatment. So,
Right. Right.
yeah.
Well, a question that you might be getting more of these days is a question about artificial intelligence, all that we're hearing about the promise of artificial intelligence, particularly in these kind of settings. So
Yeah.
we just love to get your take on what your thoughts are on this as a potential, I don't know, co-researcher, or how you see this being
Yeah,
a
so I'm going to retire in a year and a half, and I'm a dinosaur. So my own research, I'm not really focusing on AI because I think it would take me too long to learn and apply. But having said that, everybody else is really trying to scramble to use it, and you can use it for modeling. So if you've got 5 different scenarios you think might be happening, you can use AI to give you a sense as to what might be the more likely event to occur. It's being used for writing code that's being used to analyze all of these sequences. So our ability to sequence DNA 5 years ago far exceeded our ability to analyze it. So we could generate the data much more easily than we could analyze it. Now that's not the case because AI has come in and it's allowed us to do those comparisons between the healthy and the tumor DNA and find those mutations much more quickly. So there are practical applications like that. I have colleagues for whom English is not their primary language. They use it for writing, grants and correspondence. And it's not like the AI is making stuff up. They give them a template and it just puts. So there are a lot of different ways it's being used. I don't understand it enough to prognosticate as to where it's going, but anybody that's entering the field now better have a handle on it because it's going to be ubiquitous.
right.
Not
Absolutely.
a good answer. I don't know where it's going, but it's not going away.
I think that's a great answer and a very realistic, right? It's too soon to know is what it sounds like, and that's the case for a
Yeah,
lot
and
of
I have a colleague,
fields.
we had a
Yeah.
symposium a couple of weeks ago, and he's a bioinformatics guy. And I was asking him about, his use of AI, and he said, it changes week to week. And
Yeah.
so it's just such a fast moving field. It's hard to keep up with. So
Yeah.
anyway.
Well, from the questions, there is a lot of just excitement for the work that's being done, and a few of our audience members that are just concerned about federal funding cuts to research. These are stories in the news. How do we see this affecting cancer research?
It's devastating, and it's a generational gap that's gonna take 20 years to overcome. So the National Cancer Institute, which provides the bulk of the funding for work that I do and my colleagues do, has cut back on the number of grants that they fund, and the pay line is at 4%, meaning that 96% of grant submissions are not funded. 4% are. That's insane because, you know, I've sat on a number of these panel cancer grant review panels, and usually, the cream rises to the top. So you can tell the top 1%, I mean, they're clearly there, right? And you can tell the bottom 40, 50, 60%. But there's like, 20, 25% in there that could go one way or the other, and you don't know, right? And so when I started in the field, it was at 25%. Then it went down to 15%. Then it was at 10%. And now it's at 4%, which is unsustainable. The other thing is, you can tell where my political leanings are. I'm sorry, but you asked the question. We just got a grant funded to help continue to develop the cancer workforce. And we've had it for 15 years. We have to get it renewed every 5 years. The last 5-year renewal was last year. We were told that in order for it to get funded, we had to remove words from the grant such as women, diversity, inclusion, anything that would pop up a red flag if somebody did a word search of it. So we did it, we got the money, but it just burned my soul so yeah, it's really frustrating. And again, same thing with AI. I would not want to be a new or middle career investigator at this point because it's just so disheartening, even under the best of conditions. And, this is still the best job I've ever had, so I should stop whining. But under the best of conditions, 90% of the time you're banging your head against the wall. You live for the 10% of the time that it works. Now it's down to like 1% or 0.5% because of all of these limitations that are being imposed. And again, people that should be going into the field are not because they're not stupid. And that's where that big gap is going to occur. And then I'll get off the soapbox in a moment. The other big gap is that we've had a lot of foreign individuals come here to train, and then they wind up staying. They're not coming because they can't get in and they can't get, or they have to leave. And so that's a huge deficit. It's just unconscionable. Sorry.
Right. The question was asked, so no, we appreciate your
Yeah.
candid response, and you have kind of anticipated the next question, although maybe the answer is not quite what the person was looking for, but a question about advice you would give to a student entering a PhD program looking to do research on cancer immunotherapies from, the perspective of you as someone that's, looking towards retirement.
Yeah, from my my mantra has been, if you can find something you really enjoy doing and you can find out how to get paid to do it, you're better off than 95% of the people in the world. It's a very hard balance. It's a very hard combination to achieve. I've been lucky that I've been able to do that. I really love what I do. I love the outreach. This is why I talk to, OLLI. I think it's important that individuals know what we're doing. So I would argue to somebody, if this is really the field you want to get into it, because you don't want to regret. You want to minimize regrets in life, right? So when you're 40 years old, you don't want to think, God, I should have gotten into that program. You get into the program and it doesn't pan out. You're going to have a whole skill set that you can apply to a variety of different areas. So learning to be a scientist involves quite a few different skill sets: communication, critical thinking, that can be applied to a number of other areas. So if you're sure this is what you want to do, then I would still say do it. If you're unsure, you think you want to do it, work in the environment for a couple of years. It's like a laboratory technician, and see if you really like it or not. And if you don't, then that's good. You've learned something and you can get out. So I wouldn't discourage people from getting into the field, but they should get into the field with their eyes wide open.
Mhm.
And things are not permanent. So I hope the pendulum will shift again. But as I said, there's going to be a big gap that is occurring now, and so maybe that's going to be an advantage in 5 or 10 years. There you go.
Right. Absolutely. Well, as a final question, since we do have a lot of older adults in the audience with us and OLLI members, we're hearing about mutations affecting older adults more. We saw the graph of cancer rates going up as you age. Are there things older adults can do to decelerate mutations or to have any kind of effect on, reducing our cancer risk as older adults? And I want a positive, happy ending to the webinar. So don't let me down.
I mean, it's the classic stuff you hear. Your mom told you, right? Eat
Right.
eat right. Eat more vegetables. I wish I listened to my own advice, by the way. You know, eat more vegetables than donuts. Try to have incorporate some sort of activity in your life. One of probably the biggest thing is to keep your weight down, which is a hard thing to do the older you get, I realize. And then look at some of these supplements. Not a lot of them I think are still in the snake oil category, but there are some for which there is some good evidence. So moderation. I don't have a magic bullet. I wish I did, but that's that's what I can say. And unfortunately, the cancers that arise in us now started 30, 40, 50 years ago.
Mhm.
So try to keep your immune system as healthy as you can, because that's kind of the last barrier, I think.
Right, right. But enjoy a donut now and then.
Oh, yeah. Oh, as I said, everything in moderation, right?
Right,
Yeah,
exactly.
yeah.
Yeah,
So, um,
absolutely.
how novel is that? Not very, but that's the best I have. So,
Thank you. No, that's great. Well, thank you everyone for your questions. There were some questions that came in that were quite specific to specific medical conditions that I want to
yeah,
direct you to send to have a conversation with your doctor
yes.
that I did not share with the whole group. But I thank you for posting those questions as well. I want to thank you once again, Dr. Pennell, for joining us. A repeat performance and appearance on OLLI at the U. We really appreciate it. To you, the audience, for your interest, your great questions. to the OLLI program and our sponsor, the University of Minnesota Alumni Association. I hope you all will tune in next week, same time, same Zoom link, for Exploring Antarctica's Future and World Sea Level Rise by Ice Coring into the Past, which will be presented by Dr. Peter Neff, Assistant Professor of the Department of Soil, Water, and Climate at the University of Minnesota. So the learning continues. Thank you, Dr. Chris Pennell, and have a wonderful afternoon, everyone.
Thank you. Bye-bye.